Hey guys, welcome to our last topic in Chapter 20 and your last topic for the entire semester. This is going to be a really quick topic chat here, but we're going to talk about cancer. So here's our topic outline. We're going to talk about what is cancer, talk about how we identify preventable cancers, talk about the development of it really quickly, and the future of cancer treatments. As always, here's your topic objectives. Please let me know if you have any questions about these before our next exam. So let's start out with what is cancer. So as we've talked about, and we've mentioned this throughout the semester, that cancer is the uncontrolled growth of cells that then invade and colonize other sites. Those are the two key characteristics to every type of cancer. If a tumor replicates uncontrollably but doesn't spread, that's known as a benign tumor. The ones that metastasize or spread are known as metastatic, and those are the quote-unquote bad cancers because they spread and they can cause all sorts of problems. Cancer is the result of genetic instability and environmental factors. And we're not entirely sure of the amount of environmental factors that play a role, but we do know that there is that they are a contributing factor. But genetic instability is the biggest key, is that even if we were to eliminate all environmental factors, because of the genetic instability that leads to these cells that arise within the body, we will never be able to completely get rid of the formation of the cancer before it starts. So let's start with talking about preventable cancers. These are the cancers that are caused by environmental factors and have a significant component to them. For example, smoking. Smoking is a big one, or tobacco use rather. Tobacco use accounts for a significant portion of all cancer deaths yearly. And so this is an example of one that's a preventable cancer. Another example is one that is caused by an infectious disease known as HPV, and this can cause cervical cancer and throat cancers. But it's important to know that most cancers are not a result of environmental factors. So while we can point to these and say, yeah, there they are, it's not just, we just haven't found them. It's that a lot of our cancers come from internal issues, not from external causes. Now, the environment may play a role in what's causing that internally, but a lot of it is internal. And epidemiology helps us identify the differences between these because by looking at any kind of rate or any kind of rise in disease rate above endemic levels. So if it's average for 1% of the population come down with pancreatic cancer, but there's a pocket of people who have 50% of the population has pancreatic cancer, there's probably something going on there. And that's where epidemiologists come into play and start looking for these different causes to find the links to help us minimize the risks. So let's talk about how cancer actually develops in the cell, or in the body. So there's some key characteristics of cancer cells, and we've talked about these before. But the first one is the reduced reliance on external signaling. Cancer cells stop caring what their neighbors say. They're not really interested in it. They're also less likely to undergo apoptosis, which makes sense, right? Because there's two keys to developing a tumor. There has to be growth and there has to be a lack of death. And that's where you can see this on the third point, is the uncontrolled replication. Now, the cancer cells have an increased genomic mutation rate. We will see a lot of mutation within these cells. And so these cells, even though they may start as a single cell, they don't, as a tumor grows, the different cells within the tumor could have different mutations within them. Another big key is that they are invasive. They will spread from their original sites to somewhere else. And this is through that basal lamina connection being broken that we talked about earlier in this chapter. And one of the things about it is that even if cells could invade normally, they don't usually survive in that new environment. But cancer cells can. And so that's another key characteristic. And so generally speaking, they have to have multiple mutations before they become cancer cells. So we start with this one cell. And this one cell, just because it gets the one mutation, because we talked about the replication rate error from polymerase, it's not usually just one error. Now it could be, but it's not usually. Usually it takes a long time and a lot of built-up mutations within the cells to give rise to cancer. And so that's why we tend to see cancer, or why cancer tends to be an older person disease, because those cells have been around long enough to accumulate a lot more mutations. And these mutations can occur in two different types of genes that we know that are going to lead to cancer. The first is by a mutation in the oncogenes. And these genes normally are in your body, and we call them proto-oncogenes when they're not cancerous. But once they've received enough had enough mutations, they become oncogenes, which means that they're more likely to stimulate the progression towards cancer cells. Another one is tumor suppressor genes. These genes exist normally. They help prevent our cells from becoming tumors, but if they get modified, we can also see a rise and see them lead towards cancer development. So those are the two main things. And this is really brief. This one slide does not do cancer justice at all. There are entire semesters of undergraduate courses taught on how cancer works, but this is not the focus of our class. So this is the keys that I want you to understand. So let's talk a little bit about treatment. So the first key to treatment is detection. We need to detect cancer early, and the earlier we can detect it, the better. And if you think about this, this makes sense because as the cells spread throughout the body, they're going to be harder and harder to track and catch. And this is really important because if you think about it, by the time the tumor becomes palpable or maybe detectable, it's going to be pretty big already as far as how many cells are involved. We're talking 10 to the 9th cells at this point. So it's really, really important that we detect early and we find ways to screen for cancers earlier, because that means our treatments can be better. Now, one of the things that we keep talking about, and even in these last slides, we've kind of treated it this way. We've treated that cancer is one type of disease, and it's really not. Cancer as a whole has a whole lot of subsets of diseases. Even within organ sites, there's a whole lot of variety of subsets within that. So each cancer type may need its own cancer treatment, and one drug that works for one cancer type may not work for another. An excellent example of this is pancreatic cancer. Pancreatic cancer we don't have very many treatment options that work for it, and it's got a very poor survival rate, and that's because it is so unique, and it doesn't and the aspects that go along with treating it, from drug delivery to drug type to tumor type, all have to be what works normally for other cancer sites doesn't work there. And it can even be that there's different types of treatments that don't work between same tissue types. So one breast cancer drug may not work for another, and it may be individualized even. And so that's another aspect of it. So there's a whole lot of key components that go into this, and that's why treating cancer is as difficult as it is. So the three main treatment options we have are surgery, radiation, and chemotherapy, and these are what we use, but we're still trying to design better ways, more targeted methods to treat cancer and to remove it. But as you hear, is that detection is really our first line. So if we can detect it better, we may be able to treat it easier without all the damaging side effects that you see. And as I said in the last slide, this whole section could use a semester of information, and so it's really doing a disservice to just do it here on this one slide, but I wanted to make sure you got that part covered from our chapter. So this is the end of this topic. Please let me know if you have any questions, and I will see you in class.